and the generic names were pretty standard back then in the mid 70s. And when I was learning about boletes, there were basically two publications that one would use to try to figure out what it was you had in your hand or in the backyard or wherever it was you were going. And one was published by Alexander Smith from University of Michigan and Harry Tiers, my master's professor. And this was published before I started working with Harry Tiers at San Francisco. And the other publication was one that Rolf Singer started back in 1949 when he was working in Argentina. And over the years, Singer modified his concept of what boletes were based on his perception, having been in Europe, South America. And so a more conservative approach was that of Smith and Tiers, and twice the number of genera by 1986 was accepted by Rolf Singer. And I'm not saying that Smith and Tiers didn't believe what Rolf was doing or what Rolf was saying, but at the time of these publications, that's what they were looking at. And I must say that Smith and Tiers were arguing down into those things that they found in Michigan, whereas Singer had more of a global concept of what he knew about these mushrooms. But quite interestingly, back in 1972, a botanist at the University of Cambridge in England, he had been in Malaysia since the 1930s, and he collected all kinds of things, palms, mushrooms, other kinds of fungi. And then during World War II, he was under house arrest at the Singapore Botanic Garden in Singapore, and he just had to stay there. Fortunately, he was not injured or anything else. But his concept of what he had discovered in Malaysia during the time that he was there, he was very, very narrowed into what he thought the numbers of genera were. And as you can see in this slide, most everything that he had encountered, he subscribed to a subgenus rather than the genus Boletus. There's a genus Boletus, but there's all these other subgenera, right? So that was one of the big pieces of work that came out of that part of the world in the early 70s. In 1999, a student at the University of Regensburg, Manfred Binder, was the first one to actually do gene sequencing in boletes for his PhD. And I have a copy of it. It's all in German. And so it's kind of, unless you have a grasp of that language, it's kind of tough to follow. But Manfred took a postdoc at Clark University in Worcester, Massachusetts. And so Manfred and I were in communication almost daily, if not weekly. And based on the kinds of work that started with the molecular techniques using DNA sequencing, it started out very kind of touch and go, like we're just learning how to do this. So those first instances of sequencing DNA used ribosomal genes called ITS and LSU. And since the late 90s, then other more sophistication came along, and other kinds of genes were extracted and tested and sequenced. And some of those are single copy genes like GAPDH. Don't ask me what it means. It's glyceraldehyde, something, something, something. But anyway, and then there are these other protein coding genes. But there are other single copy genes in the mitochondria of cells that turned out to be helpful as well. And as I said, this was an uphill graph. So we started with the ribosomal stuff and moved towards the right in terms of accumulating data on genes. And so ultimately in 2006, Manfred Binder and David Hibbett at Clark University took gene sequences and assembled a general view of what the family Boletes, as described by Singer, and carved it up into 4 families. So things like Swillus ended up in its own family. Boletinellus ended up in its own family. Gyroporus in its own family, and Boletus in its own family, and 3 suborders. But this is all based on gene sequencing that was done in 2006. But then, as time rolled on, in and I want you to pay attention to that date there, March 2014, out of a laboratory in Kunming, China, there was a huge paper published where they sequenced over 200 different exemplars using 4 different genes, and not just Chinese collections, but other ones as well. But this was probably the tipping point of when gene data became very important in trying to understand bolete relationships. And so they described 7 clades, 7 subgroups that are now called subfamilies of the narrower concept of the family Boletacea. And as in that last line, you see that there are 59 genus level clades. And I'm going to show you a picture here of just a portion of that. And I don't want you to try to understand what's going on the left-hand side of the picture. But what on the right side, you can see clade 37, clade 39, so on and so on. They don't have any names, so they don't know at that time what they should call those genera, because everything was called Boletus. However, you can see there's a genus called Sutorius and another genus called Pulveroboletus. Those they could recognize based on the gene sequences. Those black, the generic names in black, came along within 2 months after the publication of this paper. For example, the genus Caloboletus was recognized as a genus by Alfredo Vizzini, and was published in May of the same year, 2 months later. That same year, 5 other new genera based on the phylogeny that Wu et al. Did came to pass, and those are the ones that are in black. 6 additional genera within 5 months. And this publication called Index Fungorum was a venue where you could publish online electronically. You didn't have to have a peer review. You didn't have to have photographs. You didn't have to have gene sequencing. You didn't have to, all you had to do was do it properly in terms of the code of botanical nomenclature. So you could just, boop, 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 you had to register and so on, but you could do this very, very easily. So that's what happened in late 2014. 6 more new genera were erected based on the phylogeny that the Kunming lab had presented. And let me go back. You can see the genus Neoboletus, Rubroboletus, Crocinoboletus, and Swillellus. All right, all separate genera based on the gene sequence. As it turns out, those genera describe all different red-pored boletes that were all placed in one section of Boletus, according to Smith and Tiers and Rolf Singer. So everything with red pores, according to the gene interpretations, were completely separate genera. Now, as of Friday, there are now, using that subfamily hypothesis from the Kunming lab, there are 97 genera of boletes described around the world. And one of the things about DNA sequencing and using those DNA sequences, you can find out, oh, what is this truffle-like thing? Is it a bolete? Is it an agaricus? Is it a amanita? What's it related to? It turns out that of the 97, the 21 of them are these underground truffles, and from all over the world, not just, Eastern United States or Europe. And so, as I said, as of Friday, there was one published in a paper that showed up in my doorstep on Friday that I had to add to the list here. Okay. Now, going back to before I knew anything about DNA sequencing, I had the opportunity and it was a great opportunity, to explore parts of the world where not very much was known about boletes. And I show this slide, this picture from the middle Miocene. Central America didn't exist yet. That was only 14 million years ago. Australia is still floating by itself after it broke away from Antarctica. And it hadn't run into Asia yet. That tectonic plate of Australia hadn't run into Asia yet. So Australia's been floating by itself for 40 million years. Costa Rica doesn't even exist yet until about, 9 million years ago. That's when the land bridge came between North and South America. But I had the opportunity to go to these places to see, well, what do we know about distribution, biogeography? What kinds of stuff moved into these land masses from areas that we know something about? Probably one of the most interesting spots for me was to go into northeastern Australia in the state of Queensland. And you can see there's lots of things about Queensland. It's wet, it's old, but there was this little spit of land sticking out there in the lower right. It's called Fraser Island. And it kind of intrigued me because it was designated as a World Heritage Site back in 1992. And one of the reasons for its designation was because it's the world's largest sand island. And it had rainforest. It had what the Australians would call typical bush, scrubby bush. And there are these freshwater lakes that just sit up there on top of the sand. And the only way that the lakes exist is because of rainwater. And there's just drifting sand dunes all over the place. You can see those white dots up the coast there. Those are all drifting sand dunes. And National Geographic, these are some of the sites where I was able to explore during the 3 years that I had the funding from National Geographic to explore this island. That was the other thing. All the records that I couldn't find any records that any fungi had ever been collected on this World Heritage Site. They knew all the plants, they knew all the animals, nothing about fungi. And so when I wrote the grant, I said, you don't know anything about fungi. They said, okay, what you want? And so I got a grant. So beginning in February 2009, I hooked up with a colleague at the Queensland Herbarium in Brisbane, and they were kind enough to supply a car. And my partner, Nigel, who's sitting in the driver's seat there of this four-wheel drive, and they provided the vehicle. The thing that amazed me was that we had three spare tires, not one. And I thought, where in God's name are we going where we need 3 spare tires? Well, in the background, on the skyline, in the background, on the horizon, that's the island, Fraser Island. So you have to get on, not a ferry. In Australia, it's not called a ferry. It's called a barge. You get on the barge, takes you 40 minutes to get across the straits there to get over to Fraser Island. And when you do, you get off. Fortunately, it was low tide, so we had to climb up this ramp and get on to this sand up. Now, as you all know, you drive on the left-hand side of the road in Australia on the highways. Well, you do the same thing on the beach. You drive on the left side of the beach. Fortunately, I mean, hopefully you do that when the tide's out, because if the tide's in, there's no way you're going to do that. But you see some fantastic vegetation. Typical Australian gum trees, eucalyptus. These are called scribbly barks because you look at them up close, insects just get under the bark and make these little tattoo-like things all over the surface of the bark. Growing on sand. How do trees grow on sand? They have to get their nutrients from somewhere. And it's because of the boletes that form the mycorrhizal association with the roots that the trees are able to live. And the boletes mycorrhizal hyphae, mycelium, brings the water and nutrients to the tree. The tree gives the carbon back to the boletes. But not only is there that typical, what you would call eucalypt scrub, gum scrub type bushland, there's rainforest on this sand. That's the same car that I showed you with the 3 spare tires. The main tree there, that big tree on the right, is endemic to Fraser Island and one population on the mainland of Australia. What's interesting about that tree, before, well, back in the 1800s, early 1900s, a lot of logging went on Fraser Island. That tree, or that species, is very, very adverse to accepting rotting organisms, insects, whatever. So that tree was used to help build wharfs and pylons and whatnot in the Suez Canal. And it's one of the only places in the world where that tree grows, called Syncarpia hellii. Common name, peabang. Don't ask me where that came from. I don't know. Satinay is another one. And I've seen wood, furniture made from the wood of that tree. It is absolutely gorgeous. Just a little paler than mahogany. It's beautiful stuff. So you've got rainforest growing on sand and regular scrub bushland. Oh, yeah, there's also wildlife besides trees and fungi. And because Fraser Island has been segregated from the mainland for so long, it's got probably the purest genotype of dingoes, the wild dog in Australia, on the island. They don't make a noise and they scare the bejesus out of you when you turn around and you see this dog standing behind you. And so, like they do in Yellowstone National Park, don't feed the bears. The same thing goes in Australia. Don't feed the dingoes. Put your food away, put your garbage away. But the thing that I thought interesting on this warning sign was down here. It said, if you're going bush toileting, don't go by yourself. Bring somebody with you. Now, I believe in the buddy system, but it just somehow, if I'm going to go to the John, I'm going to do it by myself. Now, I started collecting, working in Australia. I started once. I went one time in 1992, then I went back in 2005, and went once or twice a year since then, till about 2019. But over the time, I found lots of genera of boletes. I think there's 12 by 17 here. And these are all genera that are well recognized in that part of the world. And one of the really neat ones I found in 2006 up in northern Queensland. And it was a Boletus that has like a vulva, that had like an amanita. And I'm thinking, and I asked people all over the world, people that I knew, have you ever seen this thing before? anything like it. And nobody says, no, no. It turns out that with all the exploration that we've done in Queensland, we know that it grows in the far north near a town up in the north called well, we would call it Cairns. The Australians call it Cairns. And it grows all the way down to the southern border where Queensland meets New South Wales. I wanted to call it Volvo Boletes. I thought, if I did that, maybe that Swedish car company would give me some money to help me do this. Turns out that genus name had already been used, so I couldn't use Volvo Boletes. So I went with Velo Boletes. And remember I mentioned truffles being part of the bolete family? Well, we know at least 6 that grow in Australia, 7, sorry. One still isn't described yet, that one down on the lower left called Pogosperma. But the one in the upper left, Durianella, and the lower right, Spongiforma, grow in Malaysia and Indonesia. And so I put them there because it's possible they could show up in Australia, but we don't know yet. So those are truffle-like or sequestrate fungi that occur in Australia. And I show this one because I actually got to collect it more than once and prompted Jim Trappy. I said, Jim, we got to describe this thing. So he called it the sunset truffle because of its brilliant colors that reminded him of Australian sunsets. Now, as Andy mentioned, I did my PhD at UMass in Amherst working on Colibius. Well, I didn't just ignore boletes back then. There was a shoemaker in Brattleboro, Vermont. His name was Charles Frost. And his physician told him, if you're not feeling so well, maybe you should go outside a few times instead of staying in your attic and mending shoes all day. So he did, and he found a whole bunch of new boletes that he described in 1873. One of them was this iconic thing called Boletus chromopes. It has distinctive features, the pink dots on the stipe, this chrome yellow, that's how it got its species name, base to the stipe, and the pink cap. The thing I want you to recognize here is that sometimes the stipe is also has a network on it, a reticulum. It's not always just pink dots. Well, back in the 70s, when Smith and Tiers and Singer were working on boletes, Smith and Tiers had the concept, because of these physical characters, morphological characters, it belongs in the genus Tylopilus. And Singer took another route. He says, no, because of these characters, it belongs in the genus Leccinum. And depending on which way, who you wanted to follow, you'd use Tylopilus chromopes or Leccinum chromopes. And you were legitimate to do whichever you wanted. I went to Australia and I found chromopes-like thing in Australia, halfway around the world, south of the equator. I said, what? How does chromopes get there? I could not figure it out. So I said, is there an Australian Boletus chromopes? Well, 1 year later, 2 colleagues of mine, C.B. Wolf and Neil Bowser, did a paper on the chromopes group worldwide, and they adopted the use of the name that Smith and Tiers used. And so based on my collections, they described Tylopilus queenslandianus, which came from Queensland. And you can see it's got the pink dots on the stipe. They also described Tylopilus palomanus from the Paluma range in Queensland. And you can see here that the stipe has the network, the reticulum on it. But if you look closely, you can see sometimes those dots aren't just aligned with the network, they can be in between. So they describe, just because of reticulum and dots, Wolf and Bowser describe 2 separate species. Ta-da, DNA. Turns out that the Australian stuff isn't even close genetically to what we call chromopes in the United States. And so we described two new genera, Australopilus for the Australian ones, and Harria for true chromopes in the eastern United States. And it also turns out that true chromopes, it appears to be a northern hemisphere. It's also in China. And so we decided that my colleagues and I decided to name chromopes for hairy tears. We couldn't use the genus Tiersia because it had already been used before. So I said, oh, let's call it Harria for hairy tears. And so that's what we did. But based on the gene sequences that we use, here's this thing in the middle called Roy-Youngia. Yeah, what the heck is that? Turns out it's one of the truffle-like fungi that occurs in Australia, described by Jim Trappy and Mike Castellano for one of their expedition mates, a guy named Roy Young, who loved to collect truffles just for fun. That's all he liked to do for fun, was to collect truffles. It wasn't his, his rent-paying job at all in Oregon. But so Roy-Youngia fits with this chromopes group based on the genetic data that we can get. Now, let me go back to America again, come back to America again. There's this other American iconic bolete that was described from South Carolina in 1848 by Moses Ashley Curtis. And it's called the pineapple bolete. That's what ananas means, is pineapple. Because of the scaly cap, it's like the scaly parts of the fruit of a pineapple. So common term, pineapple bolete. And one of the key features that got it placed, and it was described as a Boletus, what got put into Boletus was the fact that the spores have these ribs, or sometimes called wings, on them. And you can see them there, but an interesting fact of those ribs or wings is that they have these cross bands on them that you can see with the microscope. Some people call them striae. Anyway, you see these cross bands. Well, if you're working in Australia and you're doing your homework to see what was described in Australia, and here's this thing described in 1873 called Ananaceps, so pineapple head, Boletus pineapple head. And you'll notice there's 9.5 words of Latin. That's all there is in the original description, except for spore size and a town. One of the things that amazes me is the degree of accuracy in 1873 that Reverend Miles used to describe spore length. 5 digits to the right of the decimal point. Man, I wish I had a ruler like that. Turns out, about 80 years later, if you translate the 9.5 words of Latin, it comes out, you've got this pileus with these big, thick warts all over it. That's it. You don't know what the tubes are like. You don't know what the stem or stipe is like. You don't know anything else. You don't know if it's growing in sand, soil, deadwood. You know nothing else about it. So I'm kind of going, what do we have here? Well, 80 years later, Rolf Singer looked at that original specimen from Wangaratta, and he said, spore size is different from what was originally published. And he noticed that the spores had these wings or ribs on them, but they didn't have those cross bands, those striae. And so based on what he saw in terms of the spores, he says this belongs in Boletus. Well, on Fraser Island, where we went 7 times, the first time we found that thing on the lower right, that's got those big, thick warts. The other thing that we discovered, there's some red color in there, but they're not on the warts. They're underneath and around the warts. And later on, we found young material of the same species. And we found that if you watch this thing develop, the red color is beneath this tan felty bit on top. And as the thing grows, that tan felty bit becomes those thick flocose squamose warts. And the spores are winged, ribbed, but they don't have those cross bands on them. The other thing we found out, it's got a stipe. And when you cut it open, it stains blue. But it stains blue so fast that you can't tell what color the flesh is before it stains blue, except for if you have a little cavity there, like you can see at the arrow, the flesh of the cap is yellow before it stains blue. But the blueness stays where is restricted to where the inside of the mushroom is yellow. Towards the base of the stipe, it's white and it stains brown. So just in a couple of trips to Fraser Island, we've been able to figure out and expand the concept of what the pineapple head bolete is in Australia. It's called Ananiceps. At the same time, we found another pineapple head like Boletus that didn't match anything that we knew. Turns out, while I was looking through Corner's 1972 book, Boletes in Malaysia, I came across this thing and it's doom, all of a sudden, whoa. Corner collected it in 1931 twice and twice in 1940. He didn't describe it until 1972, but he did a really good job. So it was so easy for me to figure out, hey, I've got this Boletus from Australia that matches this thing from Singapore. In Australia, it's growing with eucalyptus. In Singapore, it's growing with oak, with quercus. I thought, man, that's different. And likewise, if you cut it open, it stains blue in the cap, it's yellow fleshed, white in the stipe with a little brownish stain. But I think what was really fascinating to me was Australia's been floating by itself as a plate, tectonic plate for 40 million years. And it rams into the Asian plate. And a lot of biologists dating back to Darwin's time, Alfred Russel Wallace, noticed this discrepancy of this well, it wasn't called a barrier, but was called Wallace's line. Asian biota here, Austral biota on the other side. So I'm trying to figure out, and I have no idea what's going on, where did Discillians start? Did it start in Australia? And when Australia rammed into Asia, Ananiceps took off. Or was it the other way around? We don't know yet. So, as near as I can tell, it's the first bolete that we know about that occurs on both of Wallace's line. And to make it even more complicated, we found this other pineapple head that sort of seemed to have features of both Ananiceps and Ananas and some other things, but they were sort of blended together. So it was easy to tell apart if you had enough material in front of you. So we called it the deceptive Boletus. And we did some scanning microscopy, scanning electron microscopy of the spores. And you can see at the arrows there are these holes in the sides of the wings or ribs that go through. And I thought, wow, how come we can't see them on the surface of the ribs? I mean, that's what we can see in a light microscope. Turns out, Jacqueline Perrot in Paris in 1967 did what's called transmission electron microscopy, or she actually made sections of spores. And these spores are shown at magnifications of about 7,000 or 8,000 times. So, and inside the ribs of those spores, you can see those white spaces. Those correspond to the holes in the ribs. So what you're seeing with the light microscope, those cross bands, is not a physical bump or feature, but it's actually the result of refraction of light. When light comes up through those spores, it's refracted differently in those cavities than it is in the solid context of the spore wall. Whew, that blew me away when I read that. Now, as Andy said, I was working at the New York Botanical Garden. It's a 250-acre campus in the northern Bronx of New York. There's natural forest and there's planted, cultivated horticultural plantings of trees. But there was this very distinct bolete, another icon from the eastern United States, described by Charles Peck in 1912, based on collections from eastern Long Island. So the Bronx is about 40 miles, 60 miles, about 45 miles to the west of the eastern end of Long Island. And here's this bolete that he called Boletus balluii, named for William Ballou, who collected it. Easy to tell. You see one, you'll never forget it. But you go to Australia and you see something like balluii. But again, as I said, you're halfway around the world in another hemisphere. How is it that this species could exist also in Australia? And not only one that kind of resembles the original balluii from the eastern US, but ones that kind of take another road. They form these clusters and they have these very coarse pores and tubes. Sometimes it's almost impossible to get a spore print from one of these things because it's starting to become a gastromycete. It's starting to become a truffle or a sequestrate fungus. And that's what's happening in this particular individual here. You find them where the tubes come down onto the surface of the stipe. And they're these radially oriented tubes rather than those fine spongy-like things. And if you get up close and you look, you see pores have pores, and then those other pores have pores. So you've got 2 to 3 layers of pores in these things, totally unlike what we see with true balluii. There's some that barely even have a stipe, different color, sort of pink. There's an olive-capped one that only grows in the rainforest in northern Queensland. There's a gray-brown one with regular tubes that grows at the Kalula sandmass just south of Fraser Island. There's a white one that grows up in the Paluma Range, where Tylopilus palomanus, Australopilus palomanus was described from. And there's a yellow-orange one down near the border with New South Wales and Queensland. The only one in this group that's been described was called Phaseolasporus, and it was described from Western Australia. And that's the only place it grows. And it's a viscid one, it's sticky when it's fresh. And all of these balluis, what they have in common are these bean-shaped spores. And what puts them in the genus Tylopilus, morphologically, are these sterile cells called cystidia that have these globular oily contents, which are so easy to see with the microscope. And now, when you bring the gene application into it, there's a picture of one of those orange ones from Fraser Island on the sand. that I called Balui at the time because I didn't know what else to call it, but it fits right in there with true Tylopilus. And the reason that I have Phellius circled there is because that's the type species of the genus. And based on what the type species is, that's how we try to get concepts of other species to see if they fit in Tylopilus. So as you see, if based on gene sequencing, it hangs out right there with the type species. Now there's a whole lot of other stuff I could spend another week talking about, but I won't. But there's a lot of other newbies there in Australia that we're still trying to figure out. For example, this thing that I call Boletus lilaceps. Well, I don't know if it's a true Boletus in the sense of edgeless and the porcinis and so on. So I just use the genus name as a placeholder, but it's got a lilac cap and it stains blue as soon as you touch it, but that blue eventually turns brown. And you can't see it in the picture, but it's got a white reticulum on the stipe. In the upper right, there's this thing I call Chalciporus question mark. If you know what Boletus piperatus or Chalciporus piperatus is, we think this is related. And if so, it'll be the first report of the genus from Australia. We're still working on it. And got lots of other things, and it's in the queue, so we'll get to it sooner or later. Back in 1992, the first time I went to Queensland, I found that bolete on the lower right. And I said, this is Boletus separans, except it produced a spore print typical of Tylopilus. And when you look at the tubes in the hymenophore, that's what Tylopilus looks like. But it has the same macrochemical reaction with ammonia when you drop it on the pigmented part of the mushroom. It turns turquoise blue, just like that. And I'm thinking, oh my God, what do we have here? We have Boletus separans, but crossing boundaries in terms of morphological generic concepts. I don't know what we're doing here. So that's another one we're working on. And the last one is this thing that matches this genus called Porphyrellus. And at one time, Porphyrellus was thought to be part of Tylopilus, but it's clearly a distinctive genus. But it turns out this thing is not a Porphyrellus so far as we know, but it has, and at the time, I was calling it Porphyrellus bruneus because that was a name that was used for a New Zealand bolete, and I didn't know what else to call it. So it was a placeholder name. Then when I started doing my mycoforensics, looking at the literature, turns out that when you make a section of the pileus surface, you find these cyanogranules in the surface of the cap. And it was described as Tylopilus cyanogranulifer by Roy Watling and one of his students in 1999. As far as I know, that's the last report ever published about it. No pictures or anything. Legally and validly described as a Tylopilus. Nobody had any clue what it looked like. But that's, come on. Never mind. Anyway, turns it, come on. Uh-oh. Got a bog here. Let's try this. Of course. When we take the genes from that cyanogranulifer, we find out it's close to real Porphyrellus, Porphyriosporus there, and stroke, but it doesn't genetically, probably not either of those two genera. So we're still working on it. One of my former students is working at the Royal Botanic Garden in Melbourne. He's doing the heavy duty. So we'll try to figure out what that thing really is. So what can we take home from this? Obviously, when DNA came in as a means of trying to look at evolutionary relationships, it can help support what we already knew based on morphology, or it can alter it a little bit. There are some, more or less. Morphology still works sometimes, but not all the time. And obviously, when you travel around the world and you travel into somebody else's backyard, you always find new stuff. And that helps to get a better picture of what's going on the planet, and helps you get around this bias that we've had for a couple of centuries about what truly exists on this planet. And it's not just all northern hemisphere stuff. And lastly, I want to thank a number of people who have helped me do all of this work for the last 30 years or so. East of the Greenwich line and west of the Greenwich line, northern, southern hemisphere, lots of people. And I'll leave you with that last picture of this bolete from Australia called Gymnogaster. And one of the things that people try to do is to sequence to show that a truffle, or in this case, the reverse of a truffle, it went the other direction, that it actually belongs to another genus, Boletus, Tylopilus, Leccinum. Turns out it's been sequenced 6 times and it's still survived as a genus, Gymnogaster. Thanks so much for your listening and your patience. I appreciate it very much. Oh, yes, I don't know how to do that.
Great, so we have time for questions, and I think, can people ask questions online? They can type it into this. You shout it out. You don't have to show? No. You don't have to show.
Okay.
Here, let's change this. Yeah, why don't you use that?
Take this off, take this off. One of the former students is it's a tough thing to talk about because with boletes, you can't determine sexuality. So you can't say, this one crossed with that one, and we came out with this, because the spores don't germinate and you can't get the vegetative states to interact with one another. So it would be a tough call to say, oh, that's a hybrid. I mean, morphologically, yeah, it looks like a hybrid, but it would be the first one that I know about in the group. Does that help? Based on what I've seen of those 97 things, well, the ones that I don't know, I've seen pictures and I say, yeah, that's different. It's what's happened is that a lot of those things have shown up in places that haven't been explored before. China, Africa, Southeastern South America. And there's only, I mean, this is since 2014 with that paper from Kunming, China. That sort of set the stage, and just all of a sudden, the whole world just went nuts trying to, describe stuff, and the world opened up. People were going places nobody else had been before and describing things nobody else had seen. And with the gene tools, you're able to compare to things from the Northern Hemisphere and other stuff that's been described. So, oh yeah, and another fellow, Terry Hankel from Humboldt State, worked in Guyana in northeastern South America for 20 some years in a habitat nobody's ever looked at. And it's just been stuff like, and you look at it and you go, huh? And it's like when I first went to Australia, I just, huh? I don't get it. And the same thing. Things are showing up all over the shop. Features that you would have never thought to put together. Yes, sir.
Yeah, there's a couple of questions about your. Some question about your rock tree tree. Yeah. Okay, number one, is it aromatic? Yeah. Eucalyptus is. But it rocks. Do we know why it doesn't rot? What's its mycorrhizal cohort? Is it Boletus? So there is. Right. That is at the B-man. It was different, yeah.
Any other questions? Something comes up to.
This is a newbie question.
Sorry.
Hi. Of all these just drastic variations across the species, do you have any understanding of how much are evolutionary pressures as opposed to randomness? And kind of a follow-up is there such a thing as parallel evolution, those pressures in the kingdom? Mostly asking about what causes the variations, if there's pressures in evolution, or if a lot of these are just random, and accordingly, if there's parallel evolution across vast distances because of similar evolutionary pressure.
Great question. It's kind of a red herring for a lot of. It's a tough question to answer. There's a lot of convergent evolution, which is one of the reasons like see a lot of things like truffles showing up at various times across the tree of life. And other truffles that go on underground, turn into these occurrences. And like what Roy is demonstrating is that he's got these species that show up in the temperate, and then all of a sudden down fish island.
In Australia on the other side of the world. And they look identical from all the morphological features, nothing to differentiate them by looking at them or maybe even scoping them out. I don't know. They're probably same characteristics, but then all of a sudden you need to go to the DNA to find out that they're not related at all. But that segues into my biogeographic question I have for Roy. We have Calciporus viperatus here in the Rockies. And for those of you who will start learning, you can easily identify it. It's a bolete, got red pores. What would you imagine it tastes like if you took a bite out of it? Calciporus viperatus. It's actually peppery. It's going to be spicy. It's going to be like eating like a jalapeno, but maybe not taste as good. But you're also finding it in Australia.
That's why I had the question mark, Andy, because we're not sure. Morphologically, it looks like one, but we're still trying to get decent gene sequences to see if it matches the genus Calciporus. We don't know.
But you do get Tylopilus.
With.
And we know that is in the Northern Hemisphere and it's in Australia. So do you have any sort of like hypotheses or theories as to how that distribution came to be?
It's a tough one. It's like I pointed out with Boletus Basilians crossing Wallace's line. You know, it evolved first that type in Asia and then migrate to Australia, or did the reverse happen? So far as we know, we don't have enough data on boletes from Australia to try to tell us where things came from, where they migrated to. The only paper I know of that talks about that biogeographic hypothesis is with the genus Strobelomyces, the old man of the woods, that black scaly thing. It grows in the eastern US. And a Chinese student, I believe she was in Kunming, did this work with all the Strobelomyces she could get her hands on. And I gave her all of the ones I had collected from Australia. Based on what she could figure out, is that the genus originated in Africa, migrated to Asia, and then spread from there. And eventually trickled down into Australia. I don't know. That was her hypothesis. That's the only one I know of. Yes, sir.
More, I know they're generally mycorrhizal as far as the Boletus, but some of them are separate trophic. In Laria badia, basically, which was the new genus could be against the Xerocomus badius before. So are they starting to find more saprotrophic Boletus as well as the mycorrhizal?
So, mycorrhizal versus saprotrophic boletes.
Yes.
There are some, like the genus Boletus, is a saprotroph. It actually parasitizes a polypore. The polypore is rotting the wall. And so Boletus is saprotroph, the parasite on this polypore. So it only shows up where the polypore is on that polypore. There's another one. I don't know if you remember the slide where they had 17. And there was a picture of a person who couldn't see his face, and he was holding a bolete in front of him, and it was about that wide. It was Jim Trappy holding this thing called Boletus. It's also separate. And I forget how many species are in the genus, but it's another separate trophic. And the only time I've ever seen it in Australia is in the compact. Oh, the tree over there, Boletes over here. You know, tree roots travel a long way. Yeah.
So
So,
they're still relatively rare as compared to mycorrhizal.
yes,
Okay.
that lifestyle is minuscule. Very good. It is.
Any other questions? Last question? Well, please join me in thanking Roy. See you next month. What date is the next meeting?
April 4th,
April 4th. Do we have a speaker yet?
be announced.
We'll talk about that.
Forget
Yeah,
it.
so hopefully we'll see you next month, and thank you for coming.
Thank you.
Yeah, it's classes.
It's the t-shirt.
I am
says
too.
mushroom guy.
So I'm a research mycologist.
Okay.
And so, have you grown from spores? Yeah. So when the spore opens, hyphae comes out. The hyphae is haploid cell, just like an egg or a sperm, but it's not a male or a female. So it will only join together if it's the same species. It's called self-inclusion. When they join together, then it becomes a diploid mycelium, which can reproduce and can grow and become vegetative. So I think that's kind of what we just talked about, is that hybridization is kind of difficult.
Yeah, but
Because-
there's someone who is really often crossing. He's working with psilocybes, but he's crossing different species like on the first try all the time,
Uh-huh.
just because people haven't tried. He's doing it,
Yeah,
but I mean they're closely related species.
they are.
But-
And you know, yeah, how, if they're true species too.
Yeah,
But yeah, interesting. Yeah.
yeah. So you seem to know a lot about this.
So yeah, psilocybes is one of the easier mushrooms to grow. So
Yeah.
Yeah.
So like he did a cross between cubensis and natalensis, which are similar.
uh-huh.
He did a cross with forget what some of the other ones were, but he's like getting it on the first try. Like, he's just picking single spore isolates and crossing them, and
Which makes the question whether it's really happening or whether he's
yeah.
got contamination going on or
I should
whatever.
get your number. You seem to know
Yeah, because we
more about
when
this.
we were
than I
in
do.
college, we played around with Stropharia cubensis, and I always wondered whether it was actually psilocybe or vice versa.
Could I get your number?
Yeah. So my name is Jeff. Oh, okay.
We'll put the
Number
number
first. 719
719.
838 9619.
I already have it. I've talked to you on the phone. Okay.
What's your name?
My name is Zach. I
Zach.
called you a long
See you
time
later.
ago.
Yeah.
Because you're up in the mountains.
Yeah.
You do, like remediation. with
Yeah.
okay, I thought you-
Yeah. Okay.
Yeah, we should.
Yeah, we should. Yeah. All right. Excellent.
Cool.
Party on, man.
Yeah,
Keep growing
thanks.
mushrooms.
What's going on?
We're going.
Hi.
What's happening?
Hi, Gary.
Yeah, I have no reason.
Doing high elevation.
So,
Thank you guys.
I think I joined in December. It was my Christmas present to me. A lot of times everyone is. Yeah.